Are we set? Is our class set to be able to play on iPhone? Okay.
If you go into your integrity on here, you go to settings, then there's an option to generate a code.
Okay.
You generate your code, and then you log in onto your phone. You download the app and everything, and open the app and ask for the code, put your code in, and you go.
Okay, that's great. So you should be able to listen to these both on iPhone or on your computer if you need to. Great. So, super.
Oh, it's free? Yeah. It's a free app, too. It is.
Can't beat a free app. All right. Oh, darn. That was...
And he had his temporal lobes removed. It was temporal, correct?
Yeah, medial.
And that included a lot of his hippocampus and a lot of those structures and things like that. And in doing so, afterwards he lost his ability to create short-term memories. And so every 10, 12 seconds or so, I don't remember the exact period of time, he would forget everything that had happened. So he was perpetually, after the surgery, he would forget everything every 10 seconds. So you have to remind him of anything that had happened every 10 seconds. He was completely disabled the rest of his life. It was obviously very sad.
Yeah.
But his long-term memories. Oh, well, his long-term memories before the accident. So he would remember everything before the surgery. So he would perpetually think he was whatever age he was when he went into surgery, something like that. He couldn't. I think they found out that like some small. Did they find out that any memories could transfer at all?
Yeah, he was able to carry out procedural learning. So, for example, he could memorize strings of numbers, and he could actually carry that over without remembering that he had learned them before. So, a skill type of learning, playing the piano, things that are probably encoded in areas that don't include the part that was missing for him, the hippocampi or the medial temporal lobes of the brain. But what's really interesting and relates to microtome slicing is that H.M. did recently die and donated his body to science, and at UCSD over this past year there was a very public broadcast on the internet for 50 hours slicing of H.M.'s brain, and you can actually see, you think of very small sections of animal brains are sliced by a microtome, not too much problem. You use a paintbrush to get off this little slice and put it down on a glass dish or a glass slide, and then you can stain it. But this is a huge, very important piece of brain tissue to be slicing, and it was and they actually you could go on the web and you could see the microtome slicing H.M.'s brain, and then the slices being laid out for later analysis as well. But anyway, this is the website about H.M.'s brain, and this is a example of microtomes in action today, and what they're one of the examples of what they might be used for today. Film which shows you this process pretty nicely, hopefully. Hear that okay?
DNA makes proteins. It starts in the bundle of factors assembly at the start of the gene. It's these that trigger the first phase of the process, reading off the information that the disease makes RNA stretching into the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the DNA. It's unzipping the double helix and copying one of the 2 strands.
So it's unzipping and then it's zipping it back up, basically. So you're not seeing the unzipped portion that's within the
Standing out at the top
molecule.
is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U.
Sorry. Sorry. Don't touch it. That's what I'll remember next time.
The innermost workings of how it's simple. It's unzipping the double helix and copying one of the 2 strands of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it sneaks away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
I like that one because it gives you a sense of how quickly protein can actually be made. And of course, that's just one ribosome in one cell. In actuality, if you needed to amplify the message to create more of a protein, whether that was a hormone or a neurotransmitter that was being created, you would have many ribosomes working on that same strand of messenger RNA all in a line, polyribosomes, and you could do that in many different cells and thereby really amplify the number of protein molecules that you're creating at any time. Let's look at one more example here.
as the messenger RNA molecule is transported from the nucleus to the cytoplasm, ribosomes begin to translate the sequence into amino acids. Typically, many ribosomes translate the mRNA simultaneously. Each ribosome begins at the 5' end of the mRNA and progresses steadily towards the 3' end. New ribosomes attach to the 5' end at the same rate as the previous ones move out of the way. These multiple initiations allow the cell to make much more protein from a single message than if one ribosome had to complete the task before another could begin. When the ribosome reaches a stop codon, the ribosome and the new protein dissociate from each other and from the mRNA. This electron micrograph depicts a membrane-bound polyribosome from a eukaryotic cell.
So there you can see what was initially seen on microscopic imaging, where it just looked like a string of beads. In fact, when I learned about ribosomes, I remember my professor saying, sometimes ribosomes are all lined up like a string of beads, like they look like pearls, but we didn't know what they were doing at the time. And now we know they're all reading the same strand of messenger RNA and amplifying the message from that. That's pretty cool.
When we say in that paper and moving it to the rest of the paper
Okay, everybody ready? Close your books. From memory only, no looking. Go back to that movie that I couldn't show you because I ran out of power, and I'll just show you the parts that are most relevant to what we talked about today. Lymphocytes traveling along the endothelial cell, and this is the mechanism by which they do it. I'm actually going to skip ahead here. This is the cytoarchitecture of a flagella, actually, so neurons don't have them. But there you see an axon filament forming. There's a microtubule being formed, right? And being deconstructed at its other end. There's kinesin traveling down the microtubule and carrying its vacuole of neurotransmitter. And that's actually how it walks down the tubule. It's pretty amazing, huh? There you see the centrioles and how they're attached to the cytoskeleton. There's messenger RNA coming out of the nucleus. It's going to go and be translated into protein by ribosomes. 3 ribosomes in the cytosol. The mitochondrium. More protein synthesis. Star to show again. That's Golgi. Exocytosis could be a neurotransmitter, could be anything really. into the cell membrane, and it has these areas where it's a little less fluid. They're like floating islands. Back to the lymphocyte, rolling along the endothelial cell membrane inside it. It's going to diapedese into the tissue from the blood. Pretty cool. It must be needed. There must be damage in the tissue, so it knows to go out towards. There you go. Thanks. I'll see you guys Thursday.